demonstration on DDBJ and Uniprot databases. So, we will give details about the contents of DDBJ and how to submit the sequences and how to obtain the sequences using various search options available in DDBJ, as well as the supercomputing facility available in DDBJ. In Uniprot, we will demonstrate about the availability and the contents in Uniprot and the growth and statistics and the number of data available in Swissprot as well as TREMBL, and the different search options like simple search and advanced search options available in Uniprot, and how to extract the data and how to utilize the data for the future applications.
Hello, I am Ambuj Srivastava. Now, I am going to describe about DDBJ database, which is a nucleotide sequence database. So, you can see there are multiple options such as data submission, search and analysis, supercomputer facility and FTP thing. So, using data submission option, you can submit your sequences, nucleotide sequences into this database, and using search analysis tool you can extract the data and analyze it for future use. Then using supercomputing you can use their supercomputing facility for analyzing your data, and FTP they give some of the tools. So, first of all let me go through data submission. So, in data submission you can see there are two kind of options. One is nucleotide sequence submission system and another one is mass submission system. Then in nucleotide submission system you can, if you will go in, you can find there is a manual page. So, if you will open manual you can see. So, they have provided a link for the submission, and they also gave a step by step details of how to submit your data in DDBJ. So, apart from data submission you can also go for analysis and search. So, there are two important search options, one is get entry and another one is ARSA. So, get entry you can get entries by accession number, and in ARSA you can do all round retrieval. So, then there is taxonomic search, BLAST and different kind of search tools which you can explore further. So, here I am going to tell you about get entry and ARSA. So, first of all let me go through ARSA. So, here you can put your sequence, for example, myoglobin, and get its detail here. So, also you can take the primary accession number and copy and go to the search option using get entry. So, you can just put your ID and get the sequence in flat file format here. Also, if you want to choose a different file format, you can choose it from these options. Then in ARSA there are two kind of search options. One is quick search, another one is advanced search. So, in quick search you can directly put a keyword and get the entries. In advanced search you can choose an entry by multiple fields. For example, if you want to give a primary accession number, that will be one thing, then accession number, sequence length, molecular type and different options are available. So, for all this how you can do is that is given through an example. So, you can see if you want to match a pattern, for example, this one. So, in this pattern the last digit can be anything. So, you can see that all 5 zeros are there and then last digit is only different, otherwise everything is same. So, if you see in example, you can also do the same thing in quick search just by mentioning the field name in the start. So, PA is a primary accession number. If you will go to the quick search, which so in the same way you can do it here and you can get the same entries. So, this is all about the search and analysis options in DDBJ. Then you have a supercomputing option in the DDBJ, and how to use this supercomputing facility, this all the information you can get from this how to use the system option. So, here you can find the details. Also, if you want to transfer a file, or you want to know their hardware and software configuration, you can see it from here. So, this is all for DDBJ in this class. Now, I am going to tell you about Uniprot. So, Uniprot contains a link to lot of databases, for example, Swissprot, TREMBL, Uniref, Parks and Proteome. So, Swissprot is manually annotated and reviewed entries, then automatically annotated and not reviewed entries are there in TREMBL. Then Uniref contains sequence clusters of 3 kind, one is 50% sequence identity, another one is 90% and 100%. Then Unipark is a unique resource which contain only unique sequences, and there is no duplicacy here. And then proteomes, you get proteins of only expression proteins you can get here. So, first of all, let me take you to the about Uniprot page, where all the details of Uniprot are mentioned. For example, in the top you can see the full name of Uniprot, that is Universal Protein Resource, and which is a collaboration between EMBL, then SIB and PIR. Also, if you want to see more about how to use Uniprot KB, you can see Uniprot manual. Now, let me take you to the statistics page. So, in statistics you can get details of both kind of databases, one is Swissprot and TREMBL. So, if you will go through Swissprot, in the top option you can see there is a general detail of sequences and entries, and how the entries has been increased during the year from around 90s to now. So, then you can get the taxonomic origin details, sequence data and all other details. One important detail you can get at the end is what are the amino acid distribution in Uniprot for all these sequences. So, you can see leucine is the highest and tryptophan is lowest, as been told in the lectures. And this also could be because 6 codon code for leucine and just 1 codon code for tryptophan. Then as far as search option is concerned, you can do a simple search using keywords by writing any keyword on the top. For example, myoglobin. So, you can get the detail here for the sequences. Then if you want to filter your search further, you can do it from here. For example, if you want to check only reviewed entries, you can just click here and you will get reviewed entries. You can further refine it. For example, if you want it from human only or from mouse only, you can click somewhere around here. Or if you want to cluster your data on the basis of 100% sequence alignment or 90% or 50%, that you can do it from here. Also, only these many options are available in the first page. So, if you want to increase any column here, you can add the column and just click on the column and save. So, you can see mass column here which has been added. Now, if you want to download the data, you can click on each of the data and download it from here, or if you want to download all the data, then there is no need of selecting anyone and you can download all the data. So, in advanced search option, you can just click on advanced search and type any keyword here, or if you want it from some specific thing, you can choose from here. For example, let us say you want a sequence from human only. So, human, then let us say protein name is myoglobin, and I do not want this search criteria. So, So, in this way you can get all the sequences for myoglobin protein, in which first sequence is only reviewed, other than that all are non reviewed, which you can see from the left side, only one entry is there in reviewed part. So, this is how you can use Uniprot. Apart from that, you can do retrieve ID mapping. So, in this option what you can give is you can give an identifier from one database and identify the data from the other database. For example, if you take these two Uniprot ID and if you want to know what is the PDB IDs available for these two sequences, you can give the IDs here and choose PDB here. So, here you can see that for the first ID you have around 4 PDBs, and for the second IDs you have around 3 PDBs. So, also you can, if you want, you can perform BLAST, or if you want you can align two sequences, or by using a peptide based search also you can do. There are a lot of options available in Uniprot which you can explore from the home page. Thank you.